Hello and welcome to Chapter 29 of Biology in Focus, Resource Acquisition, Nutrition, and Transport in Vascular Plants. This is a lecture for John Tyler Community College online hybrid course. My name is Mr. Sparks and I will be your guide for this lecture. Okay, the overview in this chapter is considering the stone plants. Stone plants shown here in the left-hand corner of this photograph. Down here, they look like little animal hooves sticking up out of the ground. They're actually the succulent leaves of the plant, which exists mostly underground. Now, the photosynthetic part of the plant is basically part of the underground structure, and these leaves are actually translucent, and they allow light to pass through and to reach the photosynthetic structure, which is part of the root of this plant. So it's a plant that has a very interesting structure. You find these in the Kalahari Desert of Africa, and they're really highly adapted to the arid, high light intensity climate of that region. The success of plants depends upon their ability to gather and conserve resources from their environment. So a plant has to be able to photosynthesize, and it has to be able to store those photosynthates in its tissues. The transport of materials is central to the integrated functioning of the whole plant. So the way that plant is able to transport those photosynthates, the materials that are produced through photosynthesis, and transport them throughout the plant, and to transport water and minerals, this is all part of the integrating function of the whole plant. And the way that these function give form to each individual plant species. Adaptations for acquiring resources were key steps in the evolution of vascular plants. The evolution of adaptations enabling plants to acquire resources from both above and below the ground sources allowed for successful colonization of land by vascular plants. Okay, we remember this from a previous chapter's colonization of land by plants. The algal ancestors of land plants absorbed water, minerals, and CO2 directly from the surrounding water. Early non-vascular plants lived in shallow water and had aerial shoots that stuck up out of the water. Natural selection favored taller plants with flat appendages, multicellular branching roots, and efficient transport. Okay, these were the bryophytes, the non-vascular plants, the vascular seedless plants, the ferns, and on giving rise to the vascular seeded plants, the angiosperms and gymnosperms. The evolution of xylem and phloem in land plants made possible the development of extensive root and shoot systems that carry out long-distance transport. Xylem transports water and minerals from roots to shoots, and phloem transports photosynthetic products from sources to sink. Just think phloem food, xylem water. Okay, so here we have a schematic of a plant. It's drawing water up into the structure of the plant through the roots. Water is coming in through the roots, transporting in through the xylem of the plant structure, the tree trunk, and then the water is evaporated out through the leaves at the top of the plant. The more water that evaporates out through the leaves at the top of the plant, typically during the daytime, the more water it's able to draw up through the soil. As long as there's water in the soil, it will continue to draw up water through the plant. When there's no more water in the soil, then it's unable to draw up, and the plant may begin to wilt. Okay, carbon dioxide goes in and oxygen goes out. This is part of the photosynthetic property. So This occurs in the leaves primarily, but it also happens in the root structure. Oxygen goes into the plant, carbon dioxide leaves the plant. When carbon dioxide leaves the plant roots, it has certain effects on the chemical structure of the soil surrounding it, which enable it to incorporate cations and trace minerals into the structure of the plant. We're going to cover that in this chapter as well. Okay, so light comes in and photosynthesis and develops sugars in the leaves of the plant. The sugars travel throughout the plant. They can go in both directions. So the water goes up through the roots, out through the leaves, and the sugar is produced in the leaves, and it may go either direction depending on where there is a need. Where there's the leaves are the source of the sugars, and the other parts of the plant are could be the sink, depending on the need of the plant for the sugars and to run their metabolic functions. Adaptations in each species represent compromises between enhancing photosynthesis and minimizing water loss. So, like cactuses have very few leaves. They don't want to lose a lot of water. They have very few stoma. And then plants that live in the tropical regions, they have lots of leaves, and they have a great ability to have numerous stomata, and those stomata help it transpire and draw up water through its roots and evaporate water through its leaves. So these different adaptations in each species are compromises between photosynthesis and minimizing water loss. Shoot architecture and light capture. Stems serve as conduits of water and nutrients as supporting structures for leaves. Shoot height and branching pattern affect light capture. There is a trade-off between growing tall and branching. Okay, so generally, I think we're going to talk about this a little more in the chapter, but like in a forest, you see there's not a lot of branches where the trees are crowded together close. That's because they're all trying to reach their branches up towards the canopy where there's a greater amount of light present. When those branches on the lower ends get crowded out, they don't really receive enough light, and they will self-prune. They'll fall off that tree. So there's a trade-off between growing tall and branching, especially in a forest where you have those trees running close together. If you see a tree like an old oak tree in a field, like a cow pasture, a lot of times that tree will effectively branch out. Because all those branches can obtain sunlight, they're able to it's able to have a nice large branching tree in the middle of a field with no other trees around it. But take that same species of tree and put it in a crowded forest, and all of a sudden it loses its branches and reaches for the canopy. Phyllotaxy is the arrangement of leaves on a stem. It's specific to each species. Most angiosperms have alternate phyllotaxy with leaves arranged in a spiral. Some have paired leaves, and but phyllotaxy is the most alternate phyllotaxy is the most common. The angle between the leaves is 137.5 degrees and is likely to minimize shading of the leaves. So that phyllotaxy rotates around in a spiral. It's like, well, we'll talk about it here. Here is a gymnosperm, but it does have a good example of that radial alternate phyllotaxy. Here's the shoot apical meristem, and here is the first. This is going to be the most recent shoot, and then out here in these numbers out here, these are 40 and 42. These are the oldest shoots. So this is the newest shoot, and these are the oldest shoot. So here's the newest shoot here. Shoot 1, shoot 2, shoot 3, shoot 4, shoot 5, shoot 6, shoot 7, shoot 8, shoot 9, shoot 10, shoot 11, shoot 12, shoot 13, shoot 14. Okay, so you can see these are almost predictable in their location. Once you find 14, you know that shoot 15 is going to be catacorner across the way, 135.7 degrees, but it's shoot 15 is there, and then you'll be able to find shoot 16 because it's going to be right across the way there, and they just kind of spiral up. This is just like in that Romanesco broccoli that we looked at in the previous chapter when we talked about the mathematical patterns of the Romanesco broccoli. Well, if those patterns are uninterrupted, they will continue on like this, this alternate phyllotaxy. Okay, so that's part of the mathematical patterns of any given species of plants. The productivity of each plant is affected by the depth of the canopy, the leafy portion of the plants in the community. So a canopy of a forest is the trees that grow way up to the top. The topmost portion of the forest is the canopy, and underneath that is called the understory. And the understory is the area that you're familiar with if you go on camping trips or hikes, and it has all that vegetation in right at ground level there. That's the understory, and depending on how dense the canopy is, that's going to affect the structure of the understory. Shedding of lower shaded leaves when they respire more than photosynthesize. Self-pruning occurs when the canopy is too thick. So that's like I was talking about before, when the canopy is where the trees grow straight up tall and they have nice straight tall trunks in a forest, whereas when they grow in a field, they spread out and they grow out all over the place. So once those canopy blocks out the photosynthetic capabilities of the lower branches, then those branches will just simply fall away. It's called self-pruning. Leaf orientation affects light absorption. In low light conditions, horizontal leaves capture more sunlight. In sunny conditions, vertical leaves are less damaged by sun and allow light to reach lower leaves. Root architecture and acquisition of water and minerals. Soil is a resource mined by the root system. Root growth can adjust to local conditions. For example, roots branch more in a pocket of high nitrate than in a pocket of low nitrate. Okay, so they are able to adapt to the local conditions. They can harvest the minerals that they need based on the availability of those minerals. Roots are less competitive with other roots from the same plant than with roots from different plants. So when roots reach into a mineral resource that they require, if another plant from a different species in particular, or even another plant of the same species, is trying to obtain those resources, the first plant will then put more energy into growing those roots so that it can harvest those resources before the other plant does. Roots and hyphae of soil fungi form mutualistic associations called mycorrhizae. Okay, we covered this in the previous chapter on colonization of land by plants. Mutualisms with fungi help plants to colonize land. Mycorrhizal fungi increase the surface area for absorbing water and minerals. So mycorrhizal fungi relationships are very important and beneficial to various plant species. Most plant species have mycorrhizal fungi relationships. Different mechanisms transport substances over long or short distances. There are two major pathways through the plants, the apoplast and the symplast. The apoplast is the continuum of cell walls and extracellular spaces. So surrounding the meaty part of the cell through the cell walls, that's called apoplastic transport. Symplast is the continuum of cytosol connected by plasmodesmata. So that's the interior part of the cells that are connected by tiny little pores called plasmodesmata. The apoplast consists of everything external to the plasma membrane. It includes cell walls, extracellular spaces, and the interior vessel elements and tracheids. The symplast consists of cytosol of the living cells in a plant as well as the plasmodesmata. 3 transport routes for water and solutes are the apoplastic route through cell walls and extracellular spaces, the symplastic route through the cytosol, and the transmembrane route through cell walls. Okay, here's the apoplastic route around the interior part of the cell. It travels through the cell walls primarily. The symplastic route travels through the cytosol, through the interior regions of the cell, around all the organelles, around the nucleus, and the chloroplast might be in this area. This is the symplastic route, and the transmembrane route, which travels both the symplastic and the apoplastic route. It goes through the cytosol and then across these different membranes. Different types of nutrients travel these different pathways. Short distance transport of solutes across plasma membranes. Plasma membranes permeability controls short distance movement of substrates. Both active and passive transport occurs in plants. In plants, membrane potential is established through pumping hydrogen ions by proton pumps. In animals, membrane potential is established by pumping sodium ions through the sodium-potassium pumps. So these pumps are run, basically it's a byproduct of photosynthesis, and this is the way that the plants maintain the level of various nutrients in their cells. Okay, here's a proton pump. Let me see. We're going to go. Well, I'll go through these on this page. Here's a proton pump that's pumping. Okay, ATP is coming from the photosynthetic mechanism. It's driving the proton pump, which pumps hydrogen ions outside of the cell into the extracellular fluid. This builds up a voltage gradient. So you have negative voltage on the inside, positive voltage on the outside. And then that sets up the potential for these other capacities to exist. Okay, so hydrogen ions, and then these gates, okay, there are certain gates that allow for the transport of solutes across that. They join with these hydrogen ions. The hydrogen ions then they go through these open gates, and they take with them sucrose, for example, sugar, a certain type of sugar, or other carbohydrates. They take them across that gate and into this interior part of the cell. So you pump out the hydrogen ions, you get the positive gradient outside the cell and this extracellular fluid. The sucrose comes along. You need sucrose inside the cell, so you use hydrogen to open these gates and to pass the sucrose into the cell. Okay, it works in a similar way. Okay, so this is sucrose coming from other parts of the photosynthesis into the interior part of the cell, where then it may be used to generate ATP. Okay, nitrate is an important mineral, an important ion that can be transported the same way. You just build up a positive voltaic gradient on the outside of the cell, and nitrate ions bond with these hydrogen ions, and then they travel across this membrane gate in a similar way that sucrose does. Okay, another way to build up that positive charge on the outside of the gradient on the outside of the cell membrane is with a ion channel. Plant ion channels open and close in response to voltage, stretching the membrane and chemical factors. When open, ion channels allow specific ions to diffuse across membranes. For example, a potassium ion channel is involved in the release of potassium from guard cells when the stomata close. We'll go into that in greater detail. Okay, so here's the proton pump. It's building up hydrogen ions, positive charges on the extracellular fluid. Here's the reverse in the co-transport of cells. The neutral solutes such as sugars can be loaded into plant cells by co-transport with hydrogen ions. Hydrogen ions and sucrose co-transporters, for example, play a key role in loading sugar into the phloem prior to its transport throughout the plant. Hydrogen ions and co-transport of ions. Co-transport mechanisms involving hydrogen also participate in regulating ion fluxes into and out of the cell. For example, hydrogen co-transporters in the plasma membranes of root cells are important for the uptake of nitrate by plant roots. So this would be going on in the roots, and this would be going on in the leaves. So the leaves are loading up sucrose into the phloem, and the roots are loading up nitrates into the xylem. Okay, here's the potassium ion channel. Plant cells use the energy of hydrogen gradients to co-transport other solutes by active transport. Plant cell membranes have ion channels that allow only certain ions to pass. Short distance transport of water across plasma membranes. To survive, plants must balance water uptake and loss. Osmosis determines the net uptake or water loss by a cell and is affected by solute concentration and pressure. Water potential is the measurement that combines the effects of solute concentration and pressure. Water potential determines the direction and movement of water. Water flows from a region of higher water potential to regions of lower water potential. So an area has higher water potential if it has less solutes dissolved in it, and it has lower water potential if it has more solutes dissolved in it. So water is going to travel from the area of high water potential to the area of low water potential to try to reach equilibrium in that system. Potential refers to the water form work. Water potential is abbreviated as psi and psi, psi, and measured in a unit of pressure called the megapascal. Psi equals 0 megapascal for pure water at sea level and room temperature. Okay, so that's our control. That's our baseline is 0 megapascals of pressure at pure water at sea level at room temperature. Both pressure and solute concentration affect water potential. This is expressed by the water potential equation. Okay, psi equals psi s plus psi p, where psi s is the solute potential, psi p is the pressure potential. So the solute potential of a solution is directly proportional to have dissolved in water. The greater the molarity and the less the solute potential. So if you have let me see if I have my notes here. Okay, a 0.1 molar solution of glucose in pure water has negative 0.23 megapascal. Psi S, the solute potential, is always a negative number. Okay, so it's always going to be it's either 0 or negative or some negative number. 0 only if it's a pure water. Okay, so that solute potential, the more solutes you have in it, the solutes free water you have, and the negative, then you have a negative solute potential, increasingly negative solute potential. Solute potential is also called osmotic potential. Okay, let's see. I think there's some good information on page 576 on how solutes affect water potential. If you want more information about that subject, I'd recommend turning to that page. Pressure potential is the physical pressure on the solution. Turgor pressure is pressure exerted by the plasma membrane against the cell wall and the cell wall against the protoplast. The protoplast is the living part of the cell. So psi P is the physical pressure, could be positive or negative depending on the atmospheric pressure. Water in the tracheids at 1 atmosphere under negative pressure. Water potential affects uptake and loss by plant cells. If a flaccid lymph cell is placed in an environment with a higher solute concentration, plasmolysis. That lysis means to break apart, so the plasma will break apart. Plasmolysis occurs when the protoplast shrinks and pulls away from the cells. Now I think we have a nice diagram of this concept. Okay, so here's the initial flaccid cell in the center of the column. Psi P is indicated as 0, so that's zero pressure. It's normalized pressure all the way around, and the solute pressure is negative 0.7. So psi overall is negative 0.7 megapascals. When you put this flaccid cell into pure water, which has the pure water has more water available to it. It's got a solute pressure of 0. It allows for the plasma membrane to increase in size, because water is going in there where there's a lack of water because of the increased solute pressure. So there's a lack of water in here, so water is going from the concentration of high water through osmosis into the concentration of low water, and it's causing that membrane to expand. Conversely, if you put this solute pressure into it, okay, solute pressure of negative 0.09, that's a higher. It's inversely proportional, so it's actually a higher pressure than negative 0.7. So 0.9 is greater. So there's more solutes outside of the water here, and the water is trying to leave the plasma membrane to attain osmotic equilibrium outside of the cell. So water leaves the plasma membrane and causes the cell to go even more flaccid. Okay, this diagram is on page 577. In a flaccid cell is placed in a solution with a lower solute concentration, the cell will gain water and become turgid or firm. Turgor loss in plants causes wilting, which can be reversed when the plant is watered. Okay, so this is what's happening when your plant is wilting. It's the cells in the plant are trying to reach osmotic equilibrium, and they're expending water from inside the cells into the extracellular membrane, and it's causing it to wilt. And then you provide water for the plant. The plant takes that water up through its roots, distributes it through the xylem into the various cells, and then that increased availability of water allows those cells to become turgid once again. Okay, so here's a wilted plant versus a turgid plant. Okay, this is about the extent of my horticultural experience. You can I can tell when a plant is wilted, and I can tell when a plant is turgid. Later on in this chapter, we're going to learn about some different mineral deficiencies, and you're able to tell based on the condition of the leaf what type of mineral deficiency might be affecting that plant. So there's all kinds of details that you can understand based on the plants, but wilted versus turgid, that's about my extent of the experience. If a plant looks wilted, you can water it, and it will generally look better after that. Aquaporins facilitating diffusion of water. Aquaporins are transport proteins that affect the rate of water movement across the membrane. So it can either go quickly or slowly depending on the number of aquaporin transport proteins in that cell membrane. Okay, long distance transport, the role of bulk flow. Efficient long-distance transport of fluid requires bulk flow, the movement of a fluid driven by pressure. Water and solutes move through the tracheids and the vessel elements of the xylem and sieve tube elements of the phloem. Efficient movement is possible because mature tracheids and vessel elements have no cytoplasm, and sieve tube elements have few organelles in their cytoplasm. Plants' roots absorb essential soil. Water, air, and soil and minerals contribute to plant growth. 80 to 90% of a fresh giant oak trees in our business doing as arborists, those trees, they could weigh 10 tons. Most of that weight is water weight. 96% of the plant's dry mass consists of carbohydrates from the CO2. All of that dry mass weight, like once you remove the water from it, that dry mass weight of the tree is created out of photosynthesis through the assimilation of carbon dioxide. So it's interesting to think that all the trees that grow in the forest, as massive as they are, they're basically you can say much be correct, because only 4% of the plants think it must be deriving all of that structure from the soil, because that's where the roots are, and it does have an important role for a lot of the inorganic nutrients that are found in the soil, but most of that tree is assimilated from carbon dioxide during photosynthesis. Macronutrients and micronutrients. More than 50 chemical elements have been identified among inorganic substances in plants, but not all of these are essential to plants. There are 17 essential elements, chemical elements required for a plant to complete its life cycle. Researchers use hydroponic culture, the growth of plants in mineral solutions, to determine which chemicals are essential. So you can use this form of cultivation called hydroponics, which is where you basically grow the plant in a water solution, and you can change the variables in that water solution. It's an excellent way to perform an experiment. So you can experiment as to whether a certain nutrient is necessary by removing it from the mix. So you remove phosphorus from the mix, and you can see if that affects the growth of the plant, and versus the mix that contains all of the necessary elements. Okay, so here is an example of that. Here's the control, which is a plant receiving the solution containing all the minerals necessary, and then on to the right is the experimental solution. That is a so you could remove different compounds from this. You could take nitrogen out and see what happens to the plant then. You could take out, you want to have a control that has all the variables, and then each variable one by one you can remove those variables and see how it affects. Then you remove each variable over a period of time and measure the effect of that variable's removal on the system. Okay, it consists of all the macronutrients and micronutrients. I'm not going to go over it in too much detail here, but I do recommend that you check that out. That's on page 579. It's, carbon, oxygen, hydrogen, nitrogen, potassium, calcium. Those are the major macronutrients. The minor macronutrients are magnesium, phosphorus, and sulfur. Okay, so they're all important to the growth of the plant. And it tells you the major functions on the right. 9 of the essential elements are called macronutrients because plants require them in relatively large amounts. The macronutrients are carbon, oxygen, hydrogen, nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium. The remaining 8 are called micronutrients because plants need them in very small amounts. The micronutrients are chlorine, iron, manganese, boron, zinc, copper, nickel, and molybdenum. Plants with C4 and CAM photosynthetic pathways also need sodium. These are typically, like C4 plant would be corn, cactuses. Micronutrients function as cofactors, non-protein helpers in enzymatic reactions. Okay, so micronutrients are needed in smaller amounts, but they are important to aid in enzymatic reactions. Without the certain micronutrients, you get faulty enzymatic reactions, and it can cause problems in the growth of the plant. Symptoms of mineral deficiency depend on the nutrient's function and the mobility within the plant. Deficiency of a mobile nutrient usually affects older organs more than young ones. Deficiency of a less mobile nutrient usually affects younger organs more than older ones. The most common deficiencies are those of nitrogen, potassium, and phosphorus. So we're going to go in a little more detail, the deficiency of the mobile nutrient versus the deficiency of a less mobile nutrient. Okay, so here mineral deficiencies may vary in different species. In corn, phosphate deficient plants have reddish purple margins, particularly in the young leaves. Potassium deficient maize corn plants exhibit firing or drying along the tips or margins of the older leaves. Nitrogen deficiency is evident in the yellowing that starts at the tip and moves along the center of old leaves. Okay, so if you know the characteristics of these deficiencies, you can tell if you're like a good farmer, you can understand what's going on with your crops based on the condition of their leaves. Okay. Oh no. Well, I seem to be locked up here. So, okay, so for example, a deficiency of magnesium, a component of chlorophyll, causes chlorosis, the yellowing of the leaves. In some cases, this relationship between a mineral deficiency and its symptoms is less direct. For example, iron deficiency can cause chlorosis even if chlorophyll contains no iron, because iron ions are required as a cofactor in one of the enzymatic steps of chlorophyll synthesis. Okay, so mineral deficiency symptoms depend not only on the nutrient but also on the mobility within the plant. If a nutrient moves about freely, symptoms appear first in the older organs because young growing tissues are greater sink for nutrients than are in short supply. For example, magnesium is relatively mobile and shunted preferentially to young leaves. Therefore, a plant deficient in magnesium first shows signs of chlorosis in its older leaves. In contrast, a deficiency of a mineral that is relatively immobile affects young parts of the plant first. Older tissues may have adequate amounts that they retain during periods of short supply. For example, young leaves before any effect on the older leaves is visible. The mineral requirements of a plant can also change with the age of a plant. Young seedlings, for example, rarely show mineral deficiency symptoms because their mineral needs are met largely by the mineral reserves stored in the seeds themselves. Okay, so when we look at these different effects that it's having on the leaf. Ancient farmers recognized that crop yields would decrease on particular plot over the years. Soil management by fertilization and other practices allowed for agriculture in cities. Okay, so even like the Native Americans in this society and in this country, before the Europeans arrived, they used particular tactics to fertilize the soil when they grew their crops. They would catch fish, and they would take 2 or 3 fish and put it in a hole, and then they would plant corn on top of that, and then they would plant beans which fix nitrogen, and and squash. So they would plant corn, beans, and squash in the same hole over some dead fish, and the fish would serve as fertilizers, and the beans would serve to fix nitrogen later on in the chapter. But all these soil management has been going on since people have been growing crops on an agricultural basis, and now we have enhanced methods of using commercial fertilizers to improve the quality of the soils. In natural ecosystems, nutrients are recycled through decomposition of feces and humus, dead and organic material. Because that'd be like dumping your fish in your corn, beans, and squash. Soils can become depleted of nutrients as plants and the nutrients they contain are harvested. Fertilization replaces mineral nutrients that have been. Fertilizers are enriched in nitrogen, phosphorus, and potassium. Excess minerals are often leached from the soil and can cause algal blooms in lakes. So I think we talked about algal blooms and diatoms and red tide occurring in coastal regions like in the Gulf of Mexico. Well, these are from these excess minerals that are used in the soils in agricultural regions. So they put in, obviously you kind of want to put in more fertilizer than you need, and then that excess fertilizer just gets washed out into the creeks, and the creeks feed into the lakes, and those can cause algal blooms in the lakes, and they can kill fish if they're dense enough. Organic fertilizers are composed of manure, fish meal, or compost. Uptake the various cations, or micronutrients, is affected by the level of free hydrogen in the soil, hydrogen or pH value. Everybody remembers pH from their Bio 101 class. Cations are more available in slightly acidic soils as hydrogen ions displace mineral cations from clay particles. The availability of different minerals varies with pH. For example, a pH 8 plants can absorb calcium but not iron. At present, 30% of the world's farmland has reduced productivity. Oh, looks like we're stuck again here. Plants obtain most of their mineral nutrients from the topsoil. The basic properties of soil are texture and composition. These are like basically the qualities of the soil without considering their organic input. Soil particles are classified by size, from largest to smallest. They're called sand, silt, and clay. So clay has the smallest soil particles, and if you're all, I don't know if anybody's ever thrown a clay pot, but clay is soil that sticks together very tightly. If you've ever dug a hole in your backyard and you found you hit like a certain different horizons of soil layers, the top layer is the organic layer of humus, and below that is the B horizon where the organic layer starts to peter out, and below that you can get even into clay layers. Clay layers are formed the foundation of soil, particularly in coastal plain regions. Anyway, clay has the smallest particle size. Topsoil is formed when mineral particles released from weathered rocks mix along with living organisms and humus. So topsoil is deeper in areas where you have a lot of trees for a long period of time. So if you have an area that's been forested for thousands and thousands of years, and then you cut off those trees and you plant your soil, you plant your crops in that soil, you're going to have good soil because that's got good deep topsoil where that mineral soil has been mixed in with the humus, which is basically the fallen leaves. If you've ever, I don't know if you guys rake your own leaves in the fall, but if you fail to rake your leaves and you leave them over a year or two, they'll start to develop pockets of decomposing leaves, and those decomposing leaves are essentially humus. Okay, so that humus will build up over time. If you don't rake your lawn for years and years and years, it'll just build up and form ultimately soil. Soil solution consists of water and dissolved minerals in the pores between soil particles. After a heavy rain, water draws from the larger spaces in the soil, but smaller spaces retain water because of its attraction to clay and other particles. Loams are the most fertile topsoils. They contain equal amounts of sand, silt, and clay. A soil's composition refers to its inorganic, mineral, and organic chemical components. Inorganic components of the soil include positively charged ions or cations and negatively charged ions or anions. Most soil particles are negatively charged. Anions, for example, nitrate, let's see, they got me there. Nitrate, phosphate, and sulfate, okay, negatively charged ions such as the plant nutrients nitrate, phosphate, and sulfate do not bind with the negatively charged soil particles and are therefore easily lost by leaching and percolation of the water through the soil. Cations, for example, adhere to negatively charged soil particles, and this prevents them from leaching out of the soil and being washed away through percolating groundwater. During cation exchange, cations are displaced from soil particles by other cations. Displaced cations enter the soil solution and can be taken up by the plant roots. Okay, so here's an example. Here you've got your root hair, and your root is exuding water and carbon dioxide, and those two chemicals combine and form carbonic acid. Carbonic acid produces a negative anion, and then that joins with a positive hydrogen ion and becomes a positive cation, and then that will leap from the root hair to the soil particle and displace magnesium or potassium or calcium cations, and those cations will switch places and go into the roots. So this cation exchange only happens when there's moisture in the soil. So after there's a rain, particularly, there will be this cation exchange, and all the roots acidify the soil solution by releasing carbon dioxide from respiration and pumping hydrogen ions into the soil. Carbon dioxide reacts with water and to form carbonic acid, which releases hydrogen ion upon dissociation. The hydrogen ions in the soil neutralize the negative charge of the soil particles, causing the release of the mineral cations into the soil solution. And then the roots absorb the cations. So this is a process of the root exuding water and carbon dioxide, produce carbonic acid, which is causing the release of which releases a hydrogen ion. That hydrogen ion joins up with the soil particle and causes these cations to be released. This is called cation exchange. Organic components of the soil include decomposed leaves, feces, dead organisms, and other organic matter, which are collectively named humus. Humus forms a crumbly soil that retains water but is still porous. It also increases the soil's capacity to exchange cations and serves as a reservoir of mineral nutrients. Living components of topsoil include bacteria, fungi, algae, and other protists, including earthworms, nematodes, and plant roots. These organisms help to decompose organic material and mix it into the soil. Plant nutrition often involves relationships with other organisms. Plants and soil microbes have a mutualistic relationship. Dead plants provide energy needed by soil-dwelling microbes, and secretions from living plant roots support a wide variety of microbes in the near-root environment. Microbes have a really important role to play in plant nutrition. They help to fix nitrogen and to get nitrogen into the plant through the roots. It's a very important role that microbes play. If microbes were absent from the environment, plants would no longer be able to function, and they would be and without plants there would be no animals. So microbes are a very important part of the ecosystem. Soil bacteria exchange chemicals with plant roots, enhance decomposition, and increase nutrient availability. The soil layer surrounding the plant's roots is called the rhizosphere. The rhizobacteria thrive in the rhizosphere, and some can enter the roots. Rhizosphere has a high microbial activity because of sugars, amino acids, and organic acids that are secreted by the roots of the plant. Rhizobacteria, known as plant growth promoting rhizobacteria, can play several roles. They can produce hormones that stimulate plant growth. They produce antibiotics that protect the roots from disease, and they absorb toxic minerals or make nutrients more available to the roots. Bacteria fixation in the nitrogen cycle. Nitrogen can be an important limiting nutrient for plant growth. The nitrogen cycle transforms atmospheric nitrogen and nitrogen-containing compounds. Plants can absorb nitrogen either as nitrate, ammonium, or ammonium. The most usable soil nitrogen comes from actions of soil bacteria. Okay, so here's our plant. There's nitrogen. Nitrogen comes from the atmosphere. The atmosphere is like 80% nitrogen. It's what makes the sky blue is the light refracting off of nitrogen. It's the primary element of the atmosphere. It is mixed into the soil by the actions of bacteria, and here we can see how this nitrogen is broken down. So nitrogen comes into the soil. It's fixed as ammonium by nitrogen-fixing bacteria. There's hydrogen protons come from the soil and join with that ammonium and make it into ammonium. Now that ammonium is then directly useful by the plant. It can go into the plant root and be used and broken down into nitrates to be used by the plant. Proteins from humus and dead organic material, microbial decomposition, various amino acids, ammonifying bacteria can turn nitrogen that's been taken up by organisms and turn it into ammonium, and then it goes back into that cycle. And then alternately, ammonium can be nitrified by nitrifying bacteria. It turns it into nitrite. The nitrifying bacteria can then also turn it into nitrate, and some of that nitrate, that nitrogen is then lost into the atmosphere, and others of it, that nitrate is taken up by the plant. So nitrate and nitrogenous organic compounds are exported into the xylem, into the shoot system. So this goes in with the water, into the xylem, in through the shoot system throughout the plant. Okay, conversion to ammonium. Ammonifying bacteria break down organic compounds and release ammonium. Nitrogen-fixing bacteria converts nitrogen gas into ammonia. Yeah, I think that's right. Into ammonia. And ammonia is then converted into ammonium. Nitrifying bacteria, the conversion of conversion to nitrates. Nitrifying bacteria oxidize ammonium into nitrate through to nitrite, and then to nitrite to nitrate. NO3. Different unifying bacteria mediate each step. Nitrogen is lost to the atmosphere when denitrifying bacteria converts nitrate to nitrogen. Nitrogen is abundant in the atmosphere but unavailable to plants due to the triple bond between the atoms in nitrogen. So nitrogen is very, it's ubiquitous in the atmosphere. It's very, as we described earlier, it's the most abundant gas in the atmosphere, but that gas is not available. Nitrogen fixation is the conversion of nitrogen from nitrogen gas to ammonium. And it has to go through that phase in order to weaken those bonds and make that nitrogen available. Okay, here's the equation: nitrogen plus 8 electrons plus 8 protons, hydrogen protons, plus 16 ATP equals 2 NH3. All right, this is what the nitrogen fixing bacteria are doing. This is an equation just like cellular respiration is an equation, or photosynthesis is an equation. Some nitrogen-fixing bacteria are free-living, and others form intimate association with plant roots. Okay, some nitrogen bacteria form relationships, particularly with legumes, which are the bean family, and they form little nodules that they live inside the roots of the plant. Symbiotic relationships with nitrogen-fixing rhizobacteria provide some legumes with a source of fixed nitrogen. Along a legume's roots are swellings called nodules, composed of plant cells infected by nitrogen-fixing Rhizobium bacteria. Okay, here are the nitrogen-affected roots of the soybean plant. The spherical structures on this soybean root system are nodules containing Rhizobium bacteria. The bacteria fix nitrogen and obtain photosynthetic products made by the plants. So one thing that you can do in agriculture is you can rotate your crops between corn one year and soybeans the next year. And when you plant corn, it takes nitrogen out of the soil. There's no way that corn doesn't really fix nitrogen into the soil, but soybeans do fix nitrogen into the soil. So you plant your soybeans, and then you harvest your soybeans, and then you till in under that ground, you till in all these root nodules of the remaining from the plant, and that provides nitrogen in your soil. You can actually increase the value of your soil by depending on what type of crops that you plant. So these leguminous plants, particularly the beans, various species of beans have these relationships with these Rhizobium bacteria that will fix nitrogen in the nodules on their roots, and they become beneficial for the soil quality. Inside the root nodule, Rhizobium bacteria assume a form called bacteroids, where they contain within the vesicles formed by the root cell. The plant obtains fixed nitrogen from Rhizobium, and the plant obtains fixed nitrogen from Rhizobium, and Rhizobium obtains sugar and anaerobic environment. The legume species is associated with a particular strain of Rhizobium. Okay, now we're going to go back and talk about mycorrhizae again. We've talked about mycorrhizae in the previous two chapters, and it's an important concept because mycorrhizae are very important for plant nutrition. Mycorrhizae are mutualistic associations of fungi and roots. The fungus benefits from the steady supply of sugar from the host plant. The host plant benefits because the fungus increases the surface area for water uptake and mineral absorption. Mycorrhizal fungi also secrete growth factors that stimulate growth and branching. Mycorrhizal associations consist of two major types. Ectomycorrhizae, and you may be familiar with the word ecto means outside of, and arbuscular mycorrhizae. Arbuscular refers to the tree-like shape, like it's Arbor Day, we're going to plant trees. I'm an arborist, I cut trees. Arbuscular refers to the form of the mycorrhizae. Okay, so let's see. Here we have the ectomycorrhizae on the top form, on the top diagram. It shows the mycorrhizae are forming a relationship around the tip of the root. It forms a sheath around the root, and that mycorrhizal the ectomycorrhizae, basically it's on the outside of the rootlet, but it does penetrate in there to some degree. So it does have, there's a mantle, and then inside the root it's got fungal hyphae that grow between the cortical cells, and this helps to get nutrients and water in between the cells of the root. And then the arbuscular mycorrhizae, this is the kind that basically it invades the cells of the root, and it will go inside the cell, and then it'll start branching out, just like a little tree, just like Arbor Day, arbuscular fungi. Okay, so these little tree forms here, these little structures are called the arbuscular mycorrhizae. Okay, so here are all the parts of the root, and then this is inside the cell where the arbuscular mycorrhizae have penetrated and now and continues its growth. Okay, ectomycorrhizae. There it is. The fungus is in between the cells. Okay, ectomycorrhizae. And arbuscular mycorrhizae. This fungus is inside the cells. The fungus goes inside the cells. These are just different growth forms of different fungi. In ectomycorrhizae, the mycelium of the fungus forms a dense sheath around the surface of the root. The hyphae form a network of apoplasts, a network in the apoplast, but do not penetrate the root cells. Ectomycorrhizae occur in about 10% of plant families, most of which are woody, for example, pine, birch, and eucalyptus. In arbuscular mycorrhizae, microscopic fungal hyphae extend into the root. These mycorrhizae penetrate the cell wall but not the plasma membrane to form the branched arbusculars within the root cells. The arbuscules are important sites of nutrient transfer. Arbuscular mycorrhizae occur in about 85% of plant species, including most crops. Agricultural and ecological importance of mycorrhizae. Seeds can be inoculated with fungal spores to promote formation of mycorrhizae. Some invasive exotic plants disrupt interactions between native plants and their mycorrhizal fungi. For example, garlic mustard slows down the growth of other plants by preventing the growth of mycorrhizal fungi. I know you guys are all familiar with garlic mustard. It's very common. I think we have a picture of it here in the next slide or two. Okay, here's garlic mustard on the left. It's very common. It's not native. I'm not sure where it's from originally, but it's not native to the eastern United States. And it's an invasive exotic, and it will come into the area, and it'll destroy all the mycorrhizal relationships between the native vegetation, particularly the trees, and the mycorrhizal fungus. So here we show, here's a graph, and the top it shows the increased percent in plant biomass. Okay, you can see that the increased plant biomass, sugar maples, red maples, and white ash, it's highest in the area where the garlic mustard has not invaded. Then if you take areas of the soil where you sterilized it where it was invaded by garlic mustard, and you sterilized it where it was uninvaded, okay, you can see the growth increased in plant biomass is reduced it's reduced the same amount in the invaded area as it is in the sterilized area. So when garlic mustard invades a habitat, it's basically the same thing as sterilizing the mycorrhizal fungi of all the plants in that area. It's that effective. So it basically, what you want to look at here is comparison between these sterilized values here and the uninvaded values. So the uninvaded value, the plants are doing fine, and in the invaded values and the sterilized values, they are obviously impacted. Okay, mycorrhizal colonization. It's near zero. It's 0 for sugar maple and white ash in the invaded area, and the rate of mycorrhizal colonization in the uninvaded area is obviously much higher. It's like 30 to 40%. These are normal values. And then where the garlic mustard is, it prevents the mycorrhizae from colonizing those habitats. So it has a terrible effect on trees. Okay, here's the garlic mustard. I know you guys are all familiar with it. You may not think about it right away, but when you go out for a walk in the woods, you'll see garlic mustard, and it's very common. Like I say, it's an invasive exotic. Okay, here's these graphs again. Uninvaded ones are greater. You know, where the garlic mustard is absent, the trees do better. Where the garlic mustard is present, the trees are in trouble. Epiphytes, parasites, and carnivorous plants. Some plants have nutritional adaptations that other organisms that use other organisms in non-mutualistic ways. 3 unusual adaptations are epiphytes, parasitic plants, and carnivorous plants. Epiphytes grow on other plants and they obtain water and minerals from rain rather than tapping their host for sustenance. So epiphytes are like bromeliads, if you're familiar with bromeliads, or orchids. I know everyone knows what orchids are. Orchid plants are epiphytes. They grow on other plants and they obtain all their water and minerals from the rain rather than although they grow on their host plants, they don't tap them for resources. Okay, here's a staghorn fern, which is a tropical epiphyte. It just grows on the side of that tree. And a lot of epiphytes have a structure where they trap water. Bromeliads, like pineapple, is a bromeliad that actually grows on the ground. But the structure of the pineapple top is similar to the bromeliad structure that you find that are epiphytes that grow on trees. And these epiphytes tend to have the capacity to collect water for themselves. They have a structure that allows them to collect water and minerals from the environment. They just grow on the sides of the trees, but they don't really harvest any resources from those trees. Parasitic plants absorb water, sugars, and minerals from their living host plant. Some species photosynthesize, but others rely on host plant for sustenance. Some species parasitize the mycorrhizal hyphae of other plants. Okay, so there's mistletoe. Everybody's familiar with mistletoe. It's the Christmas tradition. You stand under the mistletoe and you got to get kissed. Well, the mistletoe is a photosynthetic parasite. It actually generates its own. Its own photosynthase. It's capable of its own photosynthesis, but it gets all of its water and some of its nutrients from the tree. They grow into the branches of the tree. You can see, if you collect them, you can see where the roots of the mistletoe grow into the tree, and they extract nutrients from the tree. So even though they're capable of living on their own, they extract nutrients from the parent tree. Okay, a dodder is a non-photosynthetic parasite. I'm not sure how this one evolved, but it grows like a tendril or a vine that grows on plants, and it will extract all of its nutrients from that plant. It can affect, I think it affects tomatoes and some other plants that you might grow around, so you have to keep an eye out for them. You got to pull them off if you find them. And then there's Indian pipe, which is a non-photosynthetic parasite of mycorrhizae. And these Indian pipe, you can actually find them in our region, in the temperate deciduous forest, in the spring, in the early summer. You'll find the Indian pipe. It produces a flower that comes above ground, and it's just pure white. It has no photosynthetic qualities at all. So it lacks chloroplasts entirely. Okay, mistletoe, dodder, Indian pipe. Carnivorous plants are photosynthetic, but they obtain nitrogen by killing and digesting mostly insects. Okay, so here are carnivorous pitcher plants. On the left here, they have the inside of this structure here. This lip is formed in such a way that if an insect steps on it, it will slip down into the inside the pitcher plant. And inside the pitcher plant, there is water and enzymes. Water comes in there from the rain, but then the pitcher plant also provides enzymes, and the enzymes will digest the insect. So the insect falls into the pitcher plant, and then it gets digested by the enzymes. Similarly, the Venus flytrap is a plant which has actual capacity for visible movement. An insect can fly into the middle of the Venus flytrap, and the Venus flytrap will close fast enough to prevent the insect from leaving, and it will then it secretes enzymes and digests the insect. Okay, and the sundew is an insect that has a little different strategy. It produces sticky resins on the tips of these stem hairs. These are trichomes, and these trichomes have like a sticky gelatinous substance on them, and an insect comes on there and lands on there and gets stuck on there, and then the plant produces enzymes that helps to digest the insects. Now, the reason that they're doing this, these carnivorous plants all live in bogs. Bogs are areas with anaerobic soils that do not have a lot of free oxygen available in them, and as such they tend not to have a lot of free ions available in that soil. So the plant is getting its nutrients, like its nitrogen, for example, instead of getting it from the soil, it's getting it from these insects. So that's how carnivorous plants are adapted to live in poor, nitrogen-poor soils. Transpiration drives the transport of water and minerals from roots to shoots via the xylem. Plants can move a large volume of water from their roots to shoots. The absorption of water and minerals by root cells. Most water and mineral absorption occurs near the root tips, where the root hairs are located and the epidermis is permeable to water. Root hairs account for much of the absorption of water by roots. After soil solution enters the roots, the extensive surface area of the cortical cell membranes enhances the uptake of water and selected minerals. The concentration of essential minerals is greater in the roots than in the soil because of active transport. The endodermis is the innermost layer of cells in the root cortex. It surrounds the vascular cylinder and is the last checkpoint for the selective passage of minerals from the cortex into the vascular tissue. Water can cross the cortex via the symplast and the apoplast. Okay, so here there's two different routes for the nutrients to enter into the root. The apoplastic route, the uptake of soil, this is route number 1, the apoplastic route. The uptake of soil solution by the hydrophilic walls of the root hairs provides access to the apoplast. Water and minerals can then diffuse into the cortex along this matrix of walls and extracellular spaces. Then, secondly, there's the symplastic route. Minerals and water that cross the plasma membrane into the root hairs can enter into the symplast. Then there's the transmembrane route. As soil solution moves along the apoplast, some water and minerals are transported into the protoplasts of the cells and the epidermis and cortex and move inward via the symplast. The endodermis controlled entry to the vascular cylinder. Within the transverse and radial walls of each endodermal cell is the Casparian strip. It's the belt of waxy material that blocks the passage of water and dissolved minerals. Only minerals already in the symplast or entering the pathway by crossing the membrane, the plasma membrane of an endodermal cell, can detour around the Casparian strip and pass into the vascular cylinder. Okay, so in your two different pathways here, in your sympastic pathway, the water solutions can go through the cell wall here and across the Casparian strip directly into the xylems, where it's then transferred to the rest of the plant. Or if it's coming through the apoplastic route, it's going to get blocked by the Casparian strip, and then it has to go enter into a cell, and then through that cell it can then cross the Casparian strip and travel into the xylem. So transport in the xylem, endodermal cells and also living cells within the vascular cylinder discharge water and minerals into their walls, the apoplast. The xylem vessels then transport water and minerals by bulk flow upward into the chute system. Okay, so there's the apoplastic route on the outside of the cells, the symplastic route through the cells. The Casparian strip blocks the apoplastic route. You have to go symplastic for a way there until you can get in. But the ultimate goal of all this water and nutrient solution is to travel into the xylem, into the plant vessels. Once it's in the vessels, it will start to move through the plant via the transpiration, where the leaves are going to draw the water up through the roots into the rest of the plant. And that's where it's going to be able to deposit its mineral contents into areas where it's most necessary. Okay, there's the Casparian strip is a little bit more in detail, shows it there. Okay, that's how the apoplastic pathway has to travel through the cell in order to get to the xylem. Water and minerals can travel into the vascular cylinder through a cortex via the apoplastic route along cell walls and extracellular spaces, the symplastic route in the cytoplasm moving through between cells through the plasmodesmata, and the transmembrane route moving from cell to cell by crossing cell membranes and cell walls. The wax Casparian strip of the endodermal wall blocks the apoplastic transfer of minerals from the cortex to this vascular cylinder. Water and minerals in the apoplast must cross the plasma membrane of an endodermal cell to enter the vascular cylinder. The endodermis regulates and transports needed minerals from the soil into the xylem. Water and minerals move from the protoplasts of the endodermal cells into their cell walls. Diffusion and active transport are involved in this movement from symphoplast to apoplast. Water and minerals now enter the tracheids and into the vessel elements. The bulk flow of transport via the xylem. Xylem sap, water and dissolved minerals, is transported from roots to leaves by bulk flow. The transport of xylem sap involves transpiration, the loss of water vapor from a plant's surface. Transpired water is replaced as water travels up from the roots. Pulling xylem sap, the cohesion-adhesion-tension hypothesis. According to the cohesion-tension hypothesis, transpiration and water cohesion pull water up from the shoots to the roots. Because remember, water has those hydrogen bonds that hold it together. You can hold, if you take the next time that you're at a restaurant, you take a straw and put it in your drink and put your finger over the end of the straw and then pull the straw out of the drink. That liquid will stay inside the straw. That's because of cohesion tension. Okay, so that water is holding itself together there. So when you take your finger off that straw, then it releases the water back into your drink. Xylem sap is normally under negative pressure or tension. So basically the reason for that is because the plant is got its stomata open and it's drawing water, evaporating water out through the leaves, and the more water that evaporates, the more water it draws in. As long as there's water in the soil, it will draw more water in. When there's not water in the soil, then it will wilt. Okay, so that xylem sap is therefore normally under negative pressure. Transpirational pull is generated when water vapor in the air spaces of the leaf diffuses down its water potential gradient and exits the leaf via the stomata. As water evaporates, the air-water interface retreats further into the mesophyll cell walls and becomes more curved. Due to the high surface tension of water, the curvature of the interface creates a negative pressure potential. This negative pressure pulls water into the xylem and into the leaf. The pulling effect results from the cohesive binding between water molecules. The transpirational pull on xylem sap is transmitted from leaves to roots. Okay, so here's a closer up understanding. Here's a cross section of the leaf. Here's the palisade mesophyll. There's the spongy mesophyll in here. There's the cuticle, the upper epidermis. Okay, all these different parts of the leaf. Here's the xylem. It's bringing water into these areas. It brings water into these air spaces. And these air spaces between the microfibrils, the parts of the spongy mesophyll, as long as there's plenty of water, that water remains stays at balance with the water pressure, the atmosphere, the pressure of the water in the surrounding content. Once that water begins to drop away, like it causes an increasing temperature. So there's more water. There's less water in the atmosphere. This draws up a lot of water from the roots. There's more water in the atmosphere. It draws up less water from the roots. And there's even more water in the atmosphere. It draws up less water from the roots. But this causes tension between the cells to increase. And as that tension increases, it draws up more water. So it kind of wants to reach this state of equilibrium back again. Okay, so at first, the water vapor is lost. Transpiration is replaced by evaporation from the water film that coats the mesophyll cells. And then up above, the evaporation from the water film causes the air-water interface to retreat further into the cell wall and become more curved. The curvature increases the surface tension and therefore the rate of transpiration. So the more that the water is needed, the more pressure it puts on it drawing up more water from the roots. Okay, so here is our water potential. Here is in the outside air is 1 megapascal. Inside the leaf, it's 7 megapascals. Inside the leaf cell walls, it's 1 megapascal. The water potential gradient goes from the ground where there is water to the air where there's a lack of water. The water is uptake from the soil through the roots. Water comes in through the soil particles into the root hair, travels through the apoplastic or the symplastic route into the tracheids, in through the tracheids into the xylem. The xylem, through cohesion, adhesion, tension theory, it moves up the trunk of the tree. Cohesion by hydrogen bonding in these individual water cells. If you remember the structure of hydrogen bonding in water from Bio 101. And then into the leaves where it travels through the xylem into the mesophyll, and the stoma are open or closed as the case may be. If they're open, they permit evaporation of the water, and that evaporation reduces the water pressure inside the cell, and that increases the cohesion adhesion, the cohesion tension that draws up more water. Okay, here's the water coming in through the roots, going up the trunk through the xylem, xylem sap into the leaf, and here's the stoma of the leaf that's open, permitting more evaporation to occur into the atmosphere, and that's drawing up more water. So the more water leaves here, the more water departs from this location, the more water is pulled up from behind. So it's like pulling up a rope through the whole tree. Cohesion and adhesion in the ascent of xylem sap. Water molecules are attracted to each other through cohesion. Cohesion makes it possible to pull a column of xylem sap. Water molecules are attracted to hydrophilic walls of xylem stem cell walls through adhesion. Adhesion of water molecules to xylem cell walls helps offset the force of gravity. Thick secondary walls prevent vessel elements and tracheids from collapsing under negative pressure. This is basically the reason why the tracheid cells are made of dead tissue. Drought, stresses, or freezes can cause cavitation in the formation of the water vapor pocket by a break in the chain of the water molecules. So you can have bubbles can form inside the xylem. Bulk flow is driven by water potential difference at the opposite ends of the xylem tissue. Bulk flow is driven by evaporation and does not require energy from the plant. Like photosynthesis, it is solar powered. Bulk flow differs from diffusion. It is different by differences in pressure potential, but not by solute potential. So you can have different amounts of solutes in the xylem sap, and it's not going to affect the way that the xylem sap is transported throughout the plant. It occurs in hollow dead cells, not across membranes of living cells. Okay, so this protects it from, the structural. It could get frozen. It could get bubbles in it. But those dead cells are going to be more resistant to structural calamity than living cells might be. It moves the entire solution, not just the water or the solutes. It is much faster than, for example, than phloem moves. Okay, 29.6. The rate of transpiration is regulated by stomata. Leaves generally have broad surface areas and high surface to volume ratios. These characteristics include increased photosynthesis and increased water loss throughout the stomata. Guard cells help balance water conservation with gas exchange for photosynthesis. About 95% of all the water a plant loses escapes through the stomata. Each stoma is flanked by a pair of guard cells, which control the diameter of the stoma by changing shape. Stomatal density is in genetic and environmental control. Mechanisms of stomatal opening and closing. Changes in turgor pressure open or close stomata. When turgid, guard cells bow outward and the pore between them opens. When flaccid, the guard cells become bowed and the pore closes. So basically, when they have a lot of water, they're open and they permit additional evapotranspiration and cohesion tension to draw more water through them. When they're short of water, they go flaccid and they close those guard cells, so protecting the water supply from evaporation. Okay. Okay, here's a guard cell surrounding a stoma, typical of an angiosperm. Inside the stoma, they have these little threads, and those threads are always putting tension on them to close. So these threads want them to close all the time. But when they have water, it creates positive pressure inside that it increases the positive pressure on the plasma membranes. Those plasma membranes basically inflate, and inflating they open up the stoma. When those plasma membranes are decreased in decreased solute, they close off. So when it has oh gosh, let me try to describe it better here. Okay, so here has here's got your water solutes. They're high. There's lots of water inside the cells here, and now there's lots of water outside the cells here. So when the water's outside the cells here, then these cells go flaccid, and then when they go flaccid, these spindle fibers close the cell doors, so they close the stoma. Changes in turgor pressure result primarily from the reversible uptake and loss of potassium ions by the guard cells. Generally, stoma open during the day and close at night to minimize water loss. Stomatal opening at dawn is triggered by light and carbon dioxide depletion. An internal clock, and there's like an internal clock inside the guard cells. All eukaryotic organisms have internal clocks. They're called circadian rhythms, are 24-hour cycles. There's also circadian rhythms that run over the year and can be calibrated based on the solstice and when the shortest day of the year is. That's when fur-bearing mammals will develop their greatest coat of fur. So these circadian rhythms run through all kinds of organisms. Drought stress can cause stomata to close during the daytime. The hormone abscisic acid is produced in response to water deficiency and causes the closure of stomata. Effects of transpiration on wilting and leaf temperature. Plants lose a large amount of water by transpiration. If the lost water is not replaced by sufficient transport of water, the plant will lose water and wilt. Transpiration also results in evaporative cooling, which can lower the temperature of a leaf and prevent protein denaturation. Adaptations that reduce evaporative water loss. Xerophytes are plants that are adapted to arid climates like cactus and succulents. Here is an interesting plant called the ocotillo. The ocotillo is during the dry part of the season in the desert, it has no leaves, but then if it rains, it may rain only for a day or two, then the ocotillo will leaf out. It'll provide itself with leaves, and then those leaves will last for as long as that plant is able to benefit from the photosynthesis, maybe a couple days or a couple of weeks, and then at the end of that period of time, it will lose those leaves again until the rains come again. And when the rains come again, it'll leaf out for a couple days, couple weeks, and then drop those leaves again. So that's a strategy for retaining water in the plant, so it doesn't lose a lot of water by evapotranspiration. Here's the old man cactus found in the Mexican desert. These are it's believed that the structure of these cactus spines helps to reduce the effect of solar radiation, because white reflects solar radiation. And okay, so these are different adaptations. Oleander, shown in the inset here, is commonly found in arid climates. Its leaves have a thick cuticle and multiple layered epidermal tissue that reduces water loss. Okay, so you can see the multiple epidermis here. There's 1, 2, 3 layers that reduce water loss. Stomata are recessed in cavities called crypts, and an adaptation to reduce the rate of transpiration by protecting the stomata from the hot, dry wind. Trichomes help minimize transpiration by breaking up the flow of air and allowing the chamber of the crypt to have a higher humidity than the surrounding atmosphere. So in these crypts right here, this is where the stoma is located, and then it's got a little structure that's built around it, a little pit, and this little pit allows for greater moisture to occur inside this area. And this is oleander. It's a desert-dwelling flower. So it has adaptations for reducing loss of moisture. Okay, here's the ocotillo without any leaves. And here it is when it's leafed out after a heavy rain. Here's the leaves of the ocotillo. You can notice something else here. They've got spines around them. Those spines are defense against animals trying to eat the leaves of the plant. Because, you know, if it has those leaves out there, it only has them for a short period of time. It needs to have that. It needs to have them protected. Here's the oleander leaf. Okay, the multiple layers of epidermal tissue and the crypt, the structure to help protect the stoma. Some desert plants complete their life cycle during the rainy season. Others have leaf modifications that reduce the rate of transpiration. Some plants have a specialized form of photosynthesis called crusulian acid metabolism, where stomatal gas exchange occurs at night. Sugars are transported from sources to sinks via the phloem. All right, I think this is our last section here. The products of photosynthesis are transported through phloem by the process of translocation. The movement of sugar from sugar sources to sugar sinks. In angiosperms, sieve tube elements are the conduits for translocation. Phloem sap is an aqueous solution that is high in sucrose. It travels from a sugar source to a sugar sink. A sugar source is an organ that is a net producer of sugar, such as mature leaves. Sugar sink is an organ that is a net consumer of sugar or storer of sugar, such as a tuber or bulb. Remember the sugar beet that we looked at in the previous chapter, and I told you all about how sugar beets are the primary source of sugar in North America? Well, the sugar beet is a sugar sink, and the leaves on top of the beet is the sugar source. A storage organ can be both a sugar sink in the summer and a sugar source in the winter. Sugar may be loaded into sieve tube elements before being exported to sinks. Depending on the species, sugar may be moved by symplastic or both symplastic and apoplastic pathways. Companion cells enhance solute movement between the apoplast and the symplast. Remember when we talk about the structure of the phloem and how the cells were connected by sieve cells, and they were living cells, but the cells that conducted all their business were called the companion cells that lived on the outside of those vessel cells. Okay, those are the companion cells. Okay. Okay, so here's a schematic. This is where your photosynthesates are coming from. In here, somewhere in the spongy mesophyll, they're being transported into the cellular mechanism of the xylem of the phloem here. Here's the vessel, xylem on top, phloem on bottom. Okay, and so these sugars that are produced by photosynthesis, photosynthetes are traveling through the cells into the through the sieve cells into the traveling into the phloem, and then once they get into the phloem, they're going to be transported throughout the rest of the cell. Okay, sucrose manufactured in mesophyll cells can travel via the symplast, the blue arrows, to the sieve tube elements. In some species, sucrose exits the symplast near sieve tubes and travels through the apoplast, the red arrows. It is then actively accumulated from the apoplast by sieve tube elements and their companion cells. Oh, let's go back here. Okay, so remember how we talk about those proton pumps and the hydrogens, and they're pumping those hydrogen ions to keep a voltaic differential here. It's negative inside and positive outside, and then you use that same gradient to force open the gates here, and those gates will transport sucrose from the inside of this cell into the phloem cell. Okay, so that's the chemiosmotic mechanism that is responsible for the active transport of sucrose, and we kind of went over this in the earlier part of this chapter. Okay, so a chemiosmotic mechanism is responsible for the active transport of sucrose into companion cells and into the sieve tube elements. Proton pumps generate a hydrogen gradient, which drives sucrose accumulation with the help of a cotransport protein that couples sucrose transport to the diffusion of hydrogen ions back into the cell. In most plants, phloem loading requires active transport. Proton pumping and co-transport of sucrose and hydrogen ions enable the cells to accumulate sucrose. At the sink, sugar molecules diffuse from the phloem to sink tissues and are followed by water. Bulk flow by positive pressure, the mechanisms of translocation in angiosperms. Phloem sap moves through the sieve tube by bulk flow, driven by positive pressure, called pressure flow. Okay, here we see the loading of sugar, the green dots, into the sieve tube at the source reduces water potential inside the sieve tube elements. This causes the tube to take up water by osmosis. Okay, so this is how we drive the more substances that we put into the water system, into the flow system, the greater the draw, the osmotic pressure. This uptake of water generates a positive pressure that forces the sap to flow along the tube. The pressure is relieved by the unloading of the sugar and the consequent loss of water at the sink. In the leaf to root translocation, xylem recycles water and from sink to source. Okay, so your water is coming from your xylem. Your xylem is traveling up here, and inside you're getting your photosynthetic cells are producing sugars, and they're depositing these sugars into the phloem. And then the more sugars that go into the phloem, the greater the osmotic pressure to force water into that system. So you get more water in there, and then you get a bulk flow. It travels it's going to travel from the area of least concentration to the area of most concentration into the area of least concentration. So up in the leaves is where you have lots of it. Down in the roots is where you don't have very much sugar. So that goes from lots of sugar to little sugar, and it keeps traveling in that direction. Okay, all the while the leaf is producing sugars, all the while the root is storing sugars. The more sugars the root stores, the more sugars get sent down by the phloem. Sometimes there are more sinks than can be supported by sources, and self-thinning is the dropping of sugar sinks, such as flowers, seeds, or fruits. Or even sometimes like branches in a tree can become a sugar sink. Like they're no longer producing enough carbohydrates, enough sugars, and so they become basically dead weight. And when they become dead weight, then they literally will be dropped off the tree. Those branches are no longer useful. Okay, this is this is on page 576, calculating and interpreting temperature coefficients, and you can do this. It's a scientific skills exercise. It just tells you how to calculate a temperature coefficient. Here are the different temperatures, and this is the percent of increase in mass of the plants due to water uptake after 30 minutes. So basically, it tells you that there's a greater, that water uptake is greater if the temperature is higher. Okay, here's our little schematic plant. It's minerals and water going in, CO2, O2, CO2 is coming out, O2 is going in. All right, that's our little planty plant. Okay, here's our root cell. Remember, there's the symplastic path through the cells, through the plasmodesmata. It's like going through a little maze here. It travels in there. There's the Casparian strip, which is constructed in such a way as it actually wraps around the cell. It doesn't go through the cell. Like, it looks like it's going through the cell here, but it doesn't do that. So it goes around the cell. So you're going through the circle here, and then the symplastic pathway into the xylem here, into where the water is transported. And the apoplastic pathway goes through the cell walls. It travels through all these cell walls. It's jumping around across here and there. Cell walls jumping here. Apoplastic pathway outside of the plasma membrane. It gets blocked by the Casparian strip. It has to jump through into the cell, and then it can pass through the cell into the where it gets taken up by the xylem. That's the pathway of water and nutrients through the root. Okay, that concludes this chapter. I know that was a pretty long chapter, and there's a lot of detail in it. Please refer to your textbook for greater detail. If you have any questions, give me a call, or if there's an emergency, like if you're having a problem with your uploading your online portion of your course, that's an emergency. Or send me an email if you just have a question. I usually answer my emails at least daily, so I'm sure I will look forward to hearing from you. All right, well, I will see you all in class. Have a good day.